Unit 4 review, cell communication and cell division. So what did we cover in class? Well, in class we talked about this idea of signal transduction. We talked about action potentials, this chemical that moves this energy across a cell. This is called a neuron. We talked about the synapse, that's at the end of a neuron. Within the neurons are neurotransmitters, and those neurotransmitters can then diffuse across this space called a synapse and cause another cell to react. And so we looked at mouse party, and we looked at how different drugs affect the neuron concentration or the uptake, or they block those neurotransmitter receptor cells. We also talked about this concept of a protein ligand or a steroid ligand. Now these ligands are what binds to these receptors to cause a reaction. Then we kind of took a cell cycle clock and mitosis view of cell communications and cell cycle. So let's talk about distances. So cells can be connected to each other, and they can have gap junctions in animal cells or plasmodesmatas. These are just openings between the cells for molecules to move across. We talked about cell-to-cell recognition later in the year with the immune response. So you can have one cell dock to another and have some reaction. And then we did some local and long distance signaling. So you have cells that can release chemicals causing some other cell to start. And we talked about that's in the hormone sections. We talked about the synaptic signaling. And finally, the long distance, where you have an endocrine cell, let's say in the hypothalamus, that then puts that releasing hormone into the blood. It goes to a target cell. That target cell then tells this process to begin and produce this hormone. So let's break it down. So you have this receptor and this molecule. And that molecule, that's the signal molecule, is called a ligand. It binds to the receptor. And in doing so, there's going to be this cascade effect, called transduction. And so let's take a look at these receptors. So this is from a G protein. And so you have these signal molecules that can bind to the receptor, or you can have this signal molecule open or close gates and allowing those ions to move in or out. So that's the receptor part. The transduction part is influenced by the environment. It responds to changes, and you have this cascade effect. And so this cascade effect is very important, and one of the molecules is called cyclic AMP. So in this picture you see the ligand binding to something called a G protein. The G protein has this cascade effect to make something called cyclic AMP. Another way to look at it is you have an inactive form becoming active, which causes another group of enzymes to become active, and then it continues and cascades down, increasing the amount of reactions taking place. So now, once that happens, that cellular response is most likely going to affect something within the nucleus, either replication, transcription and translation, or the release of molecules. So in this picture, you see the entire process, and that response activates a gene that then turns on the transcription factors, and that gene becomes active. And so we can talk about feedback mechanisms. So you can have it once that protein is produced and released, it can then bind to the receptor and stop it. Another kind of molecule that can move into the nucleus are the steroid hormones. Now these, being fat-based, move through the lipid membrane, move into the plasma and the cytoplasm, and goes directly into the nucleus to cause some transcriptional factor to turn on. Again, whatever is produced can turn around and then prevent that hormone from binding to that cell. So we have feedback. Let's turn our attention to cell cycle. Now within the cell cycle, here we're looking at a picture of mitosis, where you start with a single cell that then goes through a series of steps, prophase, metaphase, anaphase, and finally telophase, where you end up with identical cells. Now this phase of the cell cycle is the shortest part of our phase. And so you're not required to know all the steps or what happens in each of them. I'd like to focus more on that regulation of these cells. So at the end of mitosis, you get something called G1. That's the growth phase of the cell. There are checkpoints that are going to then prevent the cell from going through the next step, which is getting ready for the synthesis of DNA. That's the S phase. A secondary growth phase called G2. And then finally a checkpoint at the end of G2 that says, okay, we're now ready to begin mitosis or not. And then there's a mitosis checkpoint, a point within the system that says, okay, we should go through or maybe we should not go through. Now, you are not required to understand a little bit about CDK or something called cyclin-dependent kinases. Now, there are many different types, but the purpose here for us is to understand that it is a role in the form of a feedback mechanism so that we may regulate when a cell goes through mitosis and when a cell does not go through mitosis. So your question of the week, this unit, focuses one on signal transduction and a second one that focuses on this idea of a cell cycle regulation. But how does this connect to the mammoth story? Well, actually, it connects right here. So we're looking at the sea urchins that have been fertilized, and we're going to begin to see how they begin the process. So the little dots you see are the sperm. They've then fertilized the egg. You get this protective membrane that prevents extra sperm from entering. Then it goes through mitosis. This is called differentiation. This is when the cells begin to become specialized. Then you go from 2 to 4. If you watch the clock, you can kind of see how fast things are moving. This is called the blastocele. Blastocele has about maybe 200 to 500 cells. This is now the beginning of what is called gastrulization, or the beginning of the digestive system. So you'll notice the tube begins, and as the tube begins, you'll begin to see the beginning of the digestive tube. In this case, the mouth is forming first, and the cells are growing towards the anus. Once that tube is complete, the cells would then become active and make the neural plate, which will become the nervous system. So good luck this week. We'll see you in the Zoom.